Welcome back, everyone, and we are now going into lecture 5, where we're going to continue talking about cellular respiration. We're going to talk about the citric acid cycle, the electron transport chain, and then we're going to shift our focus to anaerobic respiration, and then we'll end our discussion on some concepts related to metabolic rate. So last day we finished talking about glycolysis, whereby we converted the glucose molecule into two pyruvate molecules. And so now we're going to talk about the citric acid cycle. And the citric acid cycle is the second part of cellular respiration. And while it doesn't use oxygen directly, it'll only occur if oxygen is present for the final stage. What I want you to know about the citric acid cycle is I want you to know all the steps of the citric acid cycle. I'd like you to know all of the products of each reaction of the citric acid cycle. But unlike glycolysis, I don't want you to worry about the enzyme names. They're all very similar. Most of them are a dehydrogenase enzyme. And you do not need to know the molecular structures of any of the compounds within the citric acid cycle. The majority of this process, as pyruvate enters this citric acid cycle, it's converted to a molecule called acetyl-CoA. And this initial conversion takes away one carbon from this. Pyruvate is a 3-carbon molecule, and one carbon is taken away in the form of carbon dioxide. And we'll see that there's also a couple of electrons here, and we'll see where those electrons go in the next image. But this pyruvate is converted to an acyl unit, or a 2-carbon unit. And when it combines with the coenzyme A, it turns into acetyl-CoA, or acetyl coenzyme A. While this goes through the citric acid cycle, there are high energy electrons that are captured in the form of NADH and FADH2. There is a little bit of ATP produced during these steps, but it's very minimal. This is the step where we get all of our carbon dioxide produced as well. So the citric acid cycle, unlike glycolysis, the citric acid cycle takes place in the mitochondria. So I'm just going to share a different screen for a moment. And so the citric acid cycle takes place in the mitochondria. So here's your cell. We'll call that a cell. And within your cell, there's the organelle, the mitochondria. So the mitochondria kind of look like this kind of shape. They're a double membrane shape. And if we zoom in on that mitochondria, I'll just highlight the main components of the mitochondria. So there's these components within it where it kind of goes in, and these little areas here are referred to. So it's a double membrane organelle, and these little projections into the actual mitochondria, these things are called the cristae of the mitochondria. And in the center of the mitochondria, there's all this fluid. So there's this fluid that exists within the mitochondria. And this fluid is called the matrix fluid. And that is where the citric acid cycle occurs. That's where the citric acid cycle occurs. It's within the matrix fluid of the mitochondria. One other thing I just want you to note, that there are two membranes here. There's an outer membrane and there's this inner membrane. And within this membrane, there's this space in between. This is referred to as the intermembrane space. So we're going to come back to this when we talk about the electron transport system. So I'll just go back to the presentation now. Okay, so the citric acid cycle consists of a whole bunch of reactions. So if there is enough oxygen, then pyruvate is transported into the mitochondria. And once it's transported into the mitochondria, this pyruvate, so it's symbolized by these three black dots, each one of these is a carbon atom. So we know that there are 3 carbon atoms within pyruvate. It's immediately converted into an acyl group, or a 2-carbon unit, or an acetyl group. And this enzyme called coenzyme A attaches to it. In the process of this pyruvate getting converted into acetyl-CoA, there is a carbon dioxide that's released. So you'll see that pyruvate goes from 3 carbons to now there's only 2 carbons, because one of these carbons is released as carbon dioxide. And in the process, there are a couple of electrons that get released as well. And these electrons are stored in the molecule NADH. So we already have an NADH molecule made. And so we're in the matrix fluid. So now this 2-carbon unit combines with acetyl-CoA. And the whole idea of acetyl-CoA is actually made from one of the B vitamins. In order to make it, the coenzyme A, you actually need one of the B vitamins called pantothenic acid, if you're interested. Anyways, this acyl unit, or this 2-carbon unit, enters the cycle. It actually enters the cycle and combines with a 4-carbon unit. So these 2 carbons from the acyl unit combine with these 4 carbons from this molecule called oxaloacetate. And when these 2 molecules come together, it makes a 6-carbon unit of what is referred to as citrate. And there's 6 carbons. Only 2 of these carbons actually come from the original glucose molecule. The other 4 are coming from oxaloacetate. So the 6-carbon citrate goes through a series of reactions. And the series of reactions, it goes from citrate to isocitrate. So this molecule gets changed slightly. And then from isocitrate, it goes to alpha-ketoglutarate. And in the process of being converted to alpha-ketoglutarate, there is another carbon dioxide released, and there's another NADH molecule produced. And then the next step is another carbon dioxide released, and another NADH molecule released. So one of the things that you can notice here is that in all instances of CO2 being released, there's an NADH molecule made. Okay, so anytime that CO2 is produced, there's also an NADH molecule made. So alpha-ketoglutarate goes to a molecule called succinyl-CoA, and succinyl-CoA gets converted to a molecule called succinate, and in the process of getting converted to succinate, it produces 1 ATP molecule. So we've now made an ATP molecule, and then succinate gets converted to fumarate, and fumarate actually is the molecule between succinate and fumarate. That's where your FADH2 molecule is made. So one of the ways that I remember that FADH2 is between succinate and fumarate is that fumarate begins with the letter F, and that's where FADH2 is made. Then from fumarate to malate, there's a water that is inserted into the reaction. And I kind of remember that because I think fire and fumes, you have to put out the fire, so you need water. That's how I remember that water is a substrate of that reaction. You get malate. Malate gets converted back into oxaloacetate. And in the process of getting converted to oxaloacetate, another NADH molecule is made. And then this oxaloacetate is ready to go through the cycle again with another acyl unit from the original glucose molecule. And this just cycles and cycles and cycles as glucose comes through. As glucose gets broken down into pyruvate and the acyl unit, this cycle just keeps happening and keeps pumping out NADH, FADH2, carbon dioxide, NADH, FADH2, carbon dioxide, and just keeps pumping it out. So if we look at our what we've produced from one pyruvate molecule, we'll see that one pyruvate molecule going through the citric acid cycle produces one FADH2 molecule, produces 4 NADH molecules, 1, 2, 3, 4. It produces 3 CO2 molecules, 1, 2, 3, and it produces 1 ATP molecule. Keep in mind that this is just for one pyruvate. You have to go through the cycle again to actually see what is produced for one glucose molecule. So if you want to know what the net result of the citric acid cycle is per glucose molecule, you have to multiply all these by 2, because each pyruvate is only half of the glucose molecule. Okay, so you would get 2 FADH2s, 8 NADH, 6 carbon dioxides, and 2 ATP. So now, all of that energy that was stored in the bonds of glucose is now stored in the high energy electrons that are within NADH and FADH2. And that's what we're going to use to make all of our ATP. And these particular reactions of the electron transport system, because we're transporting the electron through a series of proteins, this particular process occurs on those cristae of the mitochondria, on those little projections onto the inside of the cell. The process by which ATP is synthesized is something referred to as oxidative phosphorylation. It's called oxidative because oxygen is absolutely required in order to do this. And it's called phosphorylation because you are phosphorylating ADP, you're adding a phosphate to make ATP. And one thing that's absolutely essential in the production of ATP is there has to be this proton gradient. There must be a concentration gradient of hydrogen ions. And this concentration gradient is actually formed when those high energy electrons from NADH and FADH2, as they pass through these proteins, that negative charge of the electron is going to attract the positive charge of those protons, and the protons are going to get pulled through into this intermembrane space, and it's going to concentrate all of those hydrogen ions. Okay, so hydrogen ion gradient is absolutely essential. So it pulls all the protons over, and there's a very high concentration of protons in the intermembrane space. So now we'll talk about everything that is going to happen within this electron transport system. So we have NADH and FADH2. The NADH, almost all of it comes from the citric acid cycle. There's a little bit that actually comes from glycolysis as well. Remember, we made 2 NADH molecules? So some of that comes from glycolysis. So you have to transport that NADH into onto the cristae so that you can actually have those high energy electrons be transported. And so as the high energy electrons pass through the series of proteins on the inner membrane, so within the intermembrane area, it pulls hydrogen ions into the space between the 2 membranes of the mitochondria. So now you have this high concentration of protons, and this high concentration of protons is a very good source of potential energy. These protons have the potential to move back to an area of low concentration, but they're kind of stuck here right now. They don't really have a method to go through. They can't go back through these proteins, and they can't pass freely across the membrane. So they're kind of stuck, but the more protons that are produced are concentrated here, the more potential there is, the more potential energy that's stored there. in the concentration gradient between the high concentration of protons in this space and the lower concentration of protons in the matrix fluid. By the time these electrons pass through all of the proteins, and we're not going to go through the names of them, it's not really a big deal. All of the energy has been, they've given up all their energy, and the energy is now stored in this concentration gradient of protons. Each pair of electrons that goes through the electron transport chain combines with 2 hydrogen atoms and 1 oxygen atom to make water. So that's the fate of these electrons. It helps produce water. So now the question is, how do you get that potential energy converted back into an energy that can be used by the cell? Well, here's how it happens. There's another protein on the membrane of the cristae, the inner membrane of the cristae, and it's called ATP synthase. And this is the only place that hydrogen ions could pass through to go back. So hydrogen ions could pass through, and as they move through, that potential energy is now converted to energy of movement of these ions. And that potential energy now is converted to kinetic energy because it's moving across. And as it moves across, that kinetic energy is enough to allow for ADP to get phosphorylated with another phosphate to make ATP. And that kinetic energy gets captured in the bond of ATP. And so that's what ends up happening. For every, and the ratio is for every 3 protons that pass through this ATP synthase enzyme, there's 1 ATP molecule that is produced. And so again, it's that potential energy that's stored in this concentration gradient of high protons to low protons. And when that potential energy is converted to kinetic energy, that kinetic energy can get stored in the bond of the phosphate to phosphate bond of ATP. Okay, so that's how the majority of your ATP is made. So let's do a balance sheet for every glucose molecule. How much, for every glucose molecule, what is produced? So here's the balance sheet. From glycolysis, we know that we have 2 net ATP, we have 2 NADH, and we have 2 waters. From the citric acid cycle, for every glucose molecule, you get 8 NADH, you get 2 FADH2s, and you get 6 carbon dioxides and 2 ATP. And now here's what's happening for each NADH and each FADH2 molecule. For every single NADH molecule, you produce about 2.5 ATP, or 2.5 moles of ATP. For every FADH2 molecule, you make about 1.5 ATP. So that means that from the citric acid cycle, you have 8 NADH molecules. So if you multiply 8 by 2.5, you get 20 ATP for citric acid cycle. Interestingly enough, those 2 NADH from glycolysis only produced 3 ATP. And so I put a bunch of question marks there as to why does it seem like it's only 1.5 ATP for these NADH molecules. For every FADH2, there's 1.5 ATP that's made. So 2 FADH2 molecules, you get 3 ATP. So you get 2 ATP from the citric acid cycle, yields 2 ATP at the end. And those 2 net ATP from glycolysis produces 2 ATP, for a grand total of 30 net ATP for every 1 glucose molecule. So the question is, why is there only 1.5 ATP for each NADH molecule from glycolysis? And you have to remember where glycolysis occurs. It occurs in the cytosol. And in order to transport this molecule of NADH across the membrane of the mitochondria, it requires ATP. So for each NADH, you have to take away one ATP for transport. And so that's why it's only 1.5 and not 2.5. So go through that again, make sure that you understand where all the balance sheet comes from, make sure that you understand the processes of glycolysis, the citric acid cycle, and the electron transport system. But now we're going to talk about what happens when there's not enough oxygen. And the first thing that happens is the electron transport chain stops functioning. You must have oxygen in order for it to happen. And the metabolism of glucose shifts from an aerobic system to an anaerobic system. And anaerobic just means without oxygen. In this particular process, pyruvate is converted into a molecule of lactate, sometimes referred to as lactic acid. You can call it either, it's fine. And it's converted into lactate in the cytosol. And that is instead of it being transported into the mitochondria. This often happens during strenuous exercise. And the lactate that's produced is the burning sensation that you feel in your muscles when you're doing strenuous exercise. And so the main thing that happens here is that pyruvate gets converted into lactate. And when it gets converted into lactate, it uses that NADH that was made in glycolysis. And it uses those high energy electrons to produce lactate. And this particular process of converting pyruvate into lactate is catalyzed by this enzyme called lactate dehydrogenase. As a result of this anaerobic respiration that requires absolutely no oxygen, the yield for ATP for every single glucose molecule is 2. This process doesn't yield any ATP. The only ATP you get was the ATP that you got throughout glycolysis, which per glucose molecule, there's only 2 net ATP. Carbohydrates, out of the major 3 biomolecules that we can use for energy, proteins, lipids, and carbohydrates. Carbohydrates are the only ones that humans can use without the utilization of oxygen. Protein breakdown requires oxygen, and so does fat breakdown. It requires oxygen. It's absolutely required. So this is the only biomolecule that you can use as energy without the presence of oxygen. And so you can still make energy if you don't have oxygen. So when a person stops breathing, there's oxygen stored in the lungs, and there's oxygen that when it goes into the lungs and then gets transported into the bloodstream, it is bound to hemoglobin. And hemoglobin is the protein on your red blood cells, or in your red blood cells, that helps you transport oxygen to all your major organ systems. And this oxygen actually helps. It doesn't, every time that your blood cycles, it doesn't use all the oxygen. It uses about 25% of your oxygen. And so if you're not breathing, there's enough oxygen in your blood to sustain all your metabolic processes for between 2 and 3 minutes, closer to the 2-minute mark. And that actually sustains life with the aerobic processes. Even when you're not breathing for a couple of minutes, there's enough oxygen stored in your hemoglobin. After that oxygen is depleted, you can have continued life and sustain it for about another minute with anaerobic respiration without any oxygen before the demand of the cells becomes too high. And it requires more ATP. So all in all, it's anywhere between 3 and 4 minutes that you can survive without breathing. It's closer to the 3-minute mark. When you're expending energy during strenuous bursts of activity, the energy that you get is mainly derived from glycolysis and the anaerobic conversion of pyruvate to lactic acid. And so that's the burning sensation you feel in your legs after doing a bunch of leg raises with heavy weights. The reason why your muscles are quick to use this system in times of very strenuous bursts of activity is that the oxidative processes like the electron transport chain are very slow to respond, but the anaerobic process is very quick, and you can get short bursts of energy from that system. So the energy really comes from glycolysis to lactic acid, and you have an immediate, very small source of ATP that your muscles can use. So here are the possible fates of pyruvate. So you can so it went through glycolysis, you made pyruvate. If there's enough oxygen, pyruvate gets brought over to the mitochondria. It goes through the citric acid cycle and goes through the electron transport chain. If there's not enough oxygen, the pyruvate gets converted to lactate via anaerobic respiration. Talk a little bit about energy in the body and energy balance in the body. In general, we want our energy input to equal our energy output. And our energy input is really coming from diet. And it has many, many factors. Your diet is dependent on how hungry you are, how big your appetite is, how much you can tell that you're hungry, how much you can tell that you're full, or that feeling of being satiated. Diet is also determined by psychological and social factors. Energy output is determined by some unregulated heat loss. So you lose a lot of energy from heat, just from thermal regulation. But then you also use energy in any of the transport processes across membranes, mechanical work like movement, chemical work like synthesizing biomolecules for growth and maintenance, for energy storage. In an ideal world, your energy input would equal your energy output. If your energy input is greater than your energy output, that energy will end up getting stored. If your energy input is less than your energy output, you'll end up breaking down molecules in your body, either fat and or protein, in order to sustain your energy needs. Energy is usually measured in something called kilocalories, and kilocalories are denoted lowercase k, lowercase c, lowercase a, lowercase l. Notice that I'm telling you that they're all lowercase. 1 kilocalorie, and this is where it gets we're going to see how this gets a little bit confusing. 1 kilocalorie is exactly the amount of heat needed to raise the temperature of 1 kilogram of water by 1 degree Celsius. That's not where this gets confusing. It's the next slide where it gets confusing. That 1 kilocalorie is actually equal to 1 calorie, but notice that the C is capitalized. This capital C calorie is a food calorie. And so if you ever see kilocalories on your food label, it's still referring to the same number of calories with a capital C. If you look at any food label, the word calorie is capitalized, and it's capitalized on purpose because it has to be capitalized. It's a common notation on many food labels, so it might say 200 cal, but with a large C, AL. The caloric content of any food can quite easily be estimated by remembering 3 really basic numbers. The amount of energy per gram of the major biomolecules are as follows. Carbohydrates have 4 kilocalories per gram. So that would be equivalent to 4 big C calories per gram. Protein has 4 big C calories per gram, or 4 kilocalories per gram. And fat has 9 kilocalories per gram, or 9 big C calories per gram. So you can very easily calculate the um, the number of calories in a food. And this is a fairly rough estimate. So a plain bagel contains 2 grams of fat, 7 grams of protein, and 38 grams of CHO, is just the short form for carbohydrate. And so the caloric content within this is 2 grams of fat times 9 kilocalories each. So you get 18 kilocalories from each for the fat component, 7 grams of protein times 4 kilocalories each gets you 28 kilocalories, and the 38 grams of carbohydrate at 4 kilocalories each gives you 152 kilocalories. Then you just have to add them all up, and you'll get just shy of 200 calories per bagel, per plain bagel. And that's a good rough estimate for how many calories are within a bagel. Of course, it depends on the size and the thickness, and if you're just eating the bagel and you have nothing on the bagel, which is interesting. I mean, if it's a really good bagel, it's actually, I kind of prefer them without anything, but if it's not such a good bagel, you might want to throw some stuff on it. But anyways, rough estimate for how to calculate the caloric content. So now the question is, how much energy do we use? And the amount of energy that a person uses is determined by their metabolic rate. And this is just an individual's energy expenditure. Couple of values and numbers and concepts that you need to be aware of. When we talk about metabolic rate, there is a concept called basal metabolic rate, or BMR. BMR is an individual's absolute lowest metabolic rate possible. This is probably closest during the time when somebody is sleeping. You're not expending any energy, you're expending very little energy. The processes in your body are at the bare minimum. And so that would be your basal metabolic rate. It's really hard to measure accurately. So when we actually try to measure somebody's metabolic rate, we often do so we measure their metabolic rate after a 12-hour fasting period in a person who's awake but resting. So no exercise, no stress. They have to be awake but exercising. And after this 12-hour fast of being awake and resting, this is called the resting metabolic rate, or the RMR. And that's usually what would be measured in a person. We can also estimate the metabolic rate by calculating how much oxygen is used in a person. We can estimate it by looking at the amount of carbon dioxide that's produced in a person. So the more carbon dioxide that you're producing, the more biomolecules you're breaking down and the more metabolism you're going through. The more oxygen you're using, the more biomolecules you are breaking down and using. So we can actually get a ratio, and this ratio actually becomes quite important, and it'll come back to I don't want to say haunt you, but it'll come back to surprise you. Yeah, surprise is a better term. It'll come back to surprise you when we talk about the respiratory section of the course. It's only going to come back once. But this ratio of carbon dioxide to oxygen is referred to as the respiratory quotient. And so let me give you an example of this respiratory quotient. The amount of carbon dioxide you produce compared to the amount of oxygen you use depends on the composition of a person's diet. So let's assume, for example, that a person's diet is completely carbohydrate. They're only ingesting glucose. It's a very bad idea. Don't do that. You'll become deficient in a lot of things, but here's the example. So you have C6H12O6, or glucose, plus 6 molecules of oxygen you need, yields 6 molecules of carbon dioxide and 6 water molecules and a bunch of energy. Well, if we look at how much carbon dioxide is produced compared to how much oxygen is used, it's in a 1-to-1 ratio. For every 6 molecules of carbon dioxide, you need 6 molecules of oxygen. For every 6 liters of carbon dioxide you produce, you are using 6 liters of oxygen. And so the respiratory quotient in a person who has a diet consisting purely of carbohydrate, and specifically purely glucose, would be 1. That's not realistic. So that's really not realistic. So the respiratory quotient really changes depending on the combination of foods that you're ingesting. If it's purely carbohydrate, we see that it's approximately a respiratory quotient of 1. If you have a pure protein diet, your respiratory quotient goes down to about 0.8. If you only ingest fat, your respiratory quotient goes down to 0.7. The average diet, your textbook is in American, with American diet in mind. The average American diet, the respiratory quotient is 0.82. And so we sort of round that in some of our calculations to a respiratory quotient of 0.8. That's usually what we use. But what this tells us is that the average diet, if the respiratory quotient is 0.82, that on average, you'll produce 0.82 liters of carbon dioxide for every 1 liter of oxygen that's consumed. That's all that's telling you. And again, it's going to come back to surprise you, better term, surprise, in the respiratory section. And the amount of energy that's released from different foods is relatively constant, anywhere between 4.5 to 5 kilocalories of energy. There's 4.5 to 5 kilocalories of energy for every 1 liter of oxygen consumed. So knowing this information, you can actually calculate the amount of calories that somebody needs per day. So assume a 70 kilogram male consumes 430 liters of oxygen per day. And you'd get this information in a question if I asked you. You'd also get this next piece of information in the question if I asked you. Assume that 1 liter of oxygen is needed for every 4.8 kilocalories metabolized. So for every 4.8 kilocalories that you need to use, you need 1 liter of oxygen. But we know that this person needs 430 liters of oxygen every single day. And the question will be, what is the estimated metabolic rate? Well, we know that you need 430 liters of oxygen per day. And we know that the rate of metabolism is 4.8 kilocalories for every liter of oxygen. So if we multiply these numbers out, 430 liters of oxygen per day times 4.8 kilocalories per liter of oxygen, our liters of oxygen cancel out, and we get that the person will need, if we multiply these numbers together, get that the person needs approximately 2,000 kilocalories per day to maintain the oxygen requirements for normal metabolism. All the questions that I give you are going to be the same. The only things that are going to differ are the number of liters of oxygen or the amount of kilocalories that are metabolized per liter of oxygen. I'll always give you a weight in there, but notice that we didn't even have to use the weight in order to calculate the metabolic rate. So the weight is always just a red herring. You don't need to actually use it. It's for people who may not have studied this type of question or may not have got this far in the presentation and tried to write the exam and think that they have to use the weight. You don't need to use the weight. It is an extraneous piece of information. I don't have to tell you anything other than the amount of oxygen and the amount of kilocalories metabolized per liter of oxygen. Those are the only 2 pieces of information you need in order to calculate the metabolic rate. So this metabolic rate, it's a rough estimate. There are a lot of factors that influence a person's metabolic rate. And these factors include age and sex or gender. So men tend to have a metabolic rate that's slightly higher than women. When I talk about gender, I'm talking about biological gender. And so men, the reason for that is because men on average have a slightly higher percentage of lean muscle mass than women. And lean muscle mass has a higher metabolic rate because it's more metabolically active. And so it's essentially the same, but there's a slight difference in gender. Metabolic rate tends to decline with increasing age, and this declining with increasing age is as a result of the lean muscle mass declining with age as well. So as you get older, the amount of lean muscle mass that you have goes down, and so you don't have as much metabolically active tissue, and therefore your metabolic rate goes down as well. So it all really comes down to lean muscle mass, higher amounts meaning a higher metabolic rate, and adipocytes. These are just fat cells or lipid-containing cells. These are primarily composed of metabolically inactive fat droplets. And so these, if you have a higher percentage of adipocytes compared to lean muscle mass, your metabolic rate is going to be lower. Generally, weight loss programs include a couple of things. They include weight training plus aerobic exercise. And with the weight training, you're trying to increase your lean muscle mass. And increasing that lean muscle mass means that you're going to have a higher metabolic rate because you have more metabolically active tissue, and you'll have more calories that are burned because your lean muscle mass is more metabolically active. So even during aerobic exercise, if you have a higher lean muscle mass, you're going to burn more calories. Activity level, higher activity level generally responds to a higher metabolic rate. The more movement you're doing, the more muscle contraction you're having, the more energy that you need. But the more muscle contraction you're doing, the more your muscles increase in size and you increase your lean muscle mass. And so activity tends to increase the percentage of lean muscle mass. You can kind of hear a common thread here. It all comes down to lean muscle mass. So higher activity level, higher metabolic rate. Diet doesn't have as big of an effect on metabolic rate as you'd think. There is something called diet-induced thermogenesis. There's an increase in metabolic rate depending on the type of food that you ingest. If you ingest a lot of fat-containing foods, there'll be very little change in the metabolic rate. Proteins tend to have a higher change in your metabolic rate if you eat more foods that are high in protein. But this concept of diet-induced thermogenesis is that there's some theories that some foods will burn more calories to break down than you ingest, and that's not really true. The only thing that probably burns more calories and is probably next to no more calories is ice water. So you ingest ice water, your body has to do something to warm it up, so it uses some energy to warm it up, and water has 0 calories anyways, so there might be a little bit of a negative influence or a negative balance of energy when you ingest ice water, but it's so minuscule that it won't make a difference. Hormones do make a difference though. Certain hormones in your body will increase your basal metabolic rate. Good examples of these are thyroid hormone, which we're not going to talk about in this class. You may have talked about it if you took the other physiology class through Health Sci. Thyroid hormone is very important in maintaining basal metabolic rate. And if there's too much thyroid hormone, you increase your basal metabolic rate. Epinephrine is the other hormone. More epinephrine increases your metabolic rate. Genetics plays some role in basal metabolic rate and body type, but it's not as much as you'd think. So if we look at all the factors that have affected metabolic rate, a person can only influence two of them. They can influence the type and amount of physical activity, and they can influence their diet. These factors that a person can actually change are referred to as modifiable factors. All the rest of them are referred to as non-modifiable factors. You can never change your age. Um, you can't change the amount of hormone that's released in your body, things like that. So those are non-modifiable factors. So we'll finish our discussion by talking about the fates of ingested biomolecules. It really has three, there are three fates for all ingested biomolecules. It's either immediately used for energy, it's used to synthesize basic components needed for growth of cells and tissues, or if you're in a surplus, you can store the biomolecules to be available for a time when your body is in an energy deficit. And so metabolism can really be separated into two major states. You can have fed state metabolism and you can have fasted state metabolism. And the state that your body is in really dictates the metabolic processes that are most active. And so when we think about the fed state metabolism, we think more of things being stored. You have extra energy, you can store it. And the primary storage of energy in the body comes in two forms. You either store it as glycogen, which is a glucose polymer, and that's stored in both the liver as well as skeletal muscle. And then once your glycogen stores are maximized, then you store the rest of the energy in fat cells. And it's stored as lipids, and this is in your adipose tissue. And both glycogen and fat are two very compact ways that you can store energy in the body. So this next image looks a little bit intimidating, but don't let it intimidate you. It's really just a schematic looking at how the body deals with the three major biomolecules. So there's fats, there's carbohydrates, and there's proteins. And if you think about these three biomolecules as having various pools in the body, you can see how if one pool gets too high, it can spill over to another pool. The pools getting too high would mimic the state of fasted. You're in a fasted state or a fed state, and your metabolic processes are more on the storage side of things. If those pools get too low, you're in the fasted state, and you may have to start breaking down some of your body's stores of biomolecules. So just notice some of the exit pipes. We're going to focus here on glucose. So glucose, you have your glucose storage here, and glucose, these red lines indicate the normal range of glucose in the body. Normal range of glucose in the body. And you can see that when it's in the normal range, you use glucose for brain metabolism. When it's in the normal range, you'll use glucose for metabolism in most tissues. So brain metabolism and most tissues. But if you're in a state where you have too much glucose and the glucose rises above that point, you start to store the glucose as glycogen. So your glucose starts going and spilling over and getting stored to glycogen. And if it gets really high, your glucose then starts to get eliminated in the urine. That's not a good sign. That's not normal. You should never have glucose in the urine. That's a sign of disease. And so notice that as you store this glucose as glycogen, you'll eventually come to a point where your glycogen stores get full and you can no longer pump glucose in to the glycogen stores. So this glucose level is going to rise, and any extra glucose that you have is going to be brought over and stored as fat. The one thing to notice about this is that the arrows actually are indicate the direction that these biomolecules can move. And notice that there's no way to go from fat back to glucose or carbohydrates. There's no way to go from fat to protein. The only way to get rid of fat in the body is by breaking it down. Metabolism, that's the only way to get rid of fat. So continuing with glucose, as I mentioned, if glucose is way too high in the body, that is a sign of disease, and very specifically, it's a sign of disease called diabetes. And you may have heard about diabetes before. I won't talk about diabetes today, but when we discuss, when we have our section on the pancreas, I'll do a little discussion on diabetes, and we'll talk about the different types and the clinical manifestations, as well as some of the effects of untreated diabetes. So notice that no matter what the level of biomolecule is, there's really a couple of fates. You can either use the biomolecule as in metabolism, you can store the biomolecule for use later, or it can be converted to a different biomolecule. So the real question is sort of, how does the body regulate the process of taking one biomolecule and converting it to another biomolecule? How does it regulate using a biomolecule compared to storing a biomolecule? And the answer to this is in that dual control of reversible metabolic reactions that we talked about a couple of lectures ago. It's about having two different enzymes that control each direction of a metabolic reaction. In glucose metabolism, the two main players here that will help determine one direction of a reaction compared to another are insulin and glucagon. And so if we look at the control of metabolic reactions, we'll see if enzyme 1, if there's a metabolic reaction where substrate A is getting converted to product B, and there's enzyme 1 and enzyme 2, and there is equal amounts of A and B, there'll be no net synthesis of A or B. That's different if we're talking about fed state metabolism. Fed state metabolism, for example, glucose, you have a lot of glucose, you're going to convert that glucose into glycogen because you won't need all that glucose. And the main player here that regulates the conversion of glucose to glycogen is this plus sign, or in this case, this plus sign stands for insulin. So insulin is a hormone that will stimulate the production of glycogen. And so this particular reaction will occur in the direction of going from glucose to glycogen. So you're going to have net glycogen synthesis. In the opposite state, or the fasted state metabolism, you're instead of being under the influence of insulin, your body is now under the influence of the hormone glucagon. And glucagon is the particular hormone that is going to increase the rate of reaction of going from glycogen to glucose, because glucagon stimulates the breakdown of glycogen. It is a sign that your body is in a state where it needs the glucose. So you break down that glycogen. And so insulin is going to stimulate the buildup of glycogen and inhibit the breakdown of it. And glucagon is going to stimulate the breakdown of glycogen and inhibit the buildup of it. So they do opposite things. In fed state metabolism, when you're in this particular state, metabolism is generally anabolic. So you're going to build things up. The only exception to this is your basal ATP requirement. You'll always break down some glucose, and that's essential for transport in and out of cells, for nerve function, for growth and synthesis. But by and large, you're in an anabolic state. And so in fed state metabolism, the absorbed nutrients, when we're talking about nutrients, we're talking about proteins, fats, and carbohydrates. They're being used for both energy, so your basal requirements. They're being used for synthesis um, of biomolecules and for proteins and cells in your body. And then finally, they're being used for storage. So those are the fates of your nutrients when you're in a fed state metabolism. And so carbohydrates, again, will be used immediately for ATP in fed state metabolism for your basal needs. And then any carbohydrate that's not used is going to first be stored as glycogen, and then any extra is going to be stored as fat. And so here are some of the carbohydrates that or some of the fates of carbohydrates that we see in fed state metabolism. So you'll ingest glucose, and your glucose is going to be converted into glucose 6-phosphate. Some of that glucose is going to go through glycolysis. It's going to go through citric acid cycle. It's going to go through electron transport system, and you're going to make a lot of ATP for your basal needs. But in your fed state, you have too much glucose. You don't need to use it all. So you can also, your body will also take some of that glucose and convert it into glycogen. And so the majority of the glycogen is found in the liver and in the skeletal muscles. And the glycogen that is in the liver is the main source of energy for the body between meals. And so some of that glucose is going to be going through glycolysis, but the rest is going to be stored as glycogen. Eventually, your glycogen stores get full. And so I want you to notice another fate of glucose. So once your glycogen stores are full, you're actually still going to go through glycolysis. The rest of that glucose is going to go through glycolysis. It's going to go through citric acid cycle. But it's within the citric acid cycle that eventually, if you have way too much glucose going through, it ties up all of the enzymes in the citric acid cycle. And some of that glucose, or some of that partially broken down glucose, is now kicked over into the fat synthesis side of things. So the next slide is about building up fat. So any extra glucose will go through this process. You'll end up going through the beginning of the citric acid cycle. And then if there's way too much going through the citric acid cycle, the rest is stored as fat. And again, there's no way of going backwards from fat into carbohydrate. So notice the reversal arrows. You can go from fat into acetyl-CoA, but you actually can't go back to pyruvate. You can't go backward to pyruvate and glucose. There's no enzyme that allows for the full conversion of fatty acids back, or the broken down units of fatty acids back into glucose. So once you're in a state where you're storing fat, if you're ever in a state where you now break down that fat, the fat will get broken down, and it's required to go through the citric acid cycle and electron transport system to be oxidized fully. That's the only way to get rid of fat in the body, is to go through oxidative phosphorylation. Okay, and so this is just a summary slide that in the fed state metabolism, you get glycogen synthesis and you get fat synthesis, as well as some of your basal needs of glucose going through cellular respiration to make your basal amount of ATP that you need. So we'll end there today. I know that that was a lot of information, but that ends our discussion on metabolism. And so next day we'll start on the GI system. And when we discuss the GI system, we're always going to start each section by going through a little bit of the relevant anatomy first, so that we build a basis for the structures that we're going to be talking about, so that you can, in your mind, you can link the structure with the function.